Hi, my name is Stephan Kadauke, and I'm an assistant professor of pathology and lab medicine at the Children's Hospital of Philadelphia, or CHOP. And at CHOP, I co-direct the cell and gene therapy lab. So today I'm going to talk about chimeric antigen receptor T cells, or CAR-T cells. And I want to highlight the role of the CHOP clinical labs in providing CAR-T cell therapy to CHOP patients. So here's the outline of my talk today. I'm going to start by introducing the CHOP Cell and Gene Therapy Lab, or the CGTL. Then I'm going to provide some background on leukemia in children. Then I'm going to answer the question, what is a CAR-T cell? And then we're going to talk about CD19-directed CAR-T cell therapy and manufacturing of such CAR-T cells. Then I want to talk briefly about a clinical trial of CAR-T cells that are being manufactured in our cell and gene therapy lab. And I want to end the session by talking a little bit about release testing for CAR-T cell products, which are critical FDA mandated tests that need to be finished before the product can be released and infused to patients. So here you can see the blueprints of the CHOP Cell and Gene Therapy Lab. This is a facility in the main building of the Children's Hospital, and the CGTL is accredited by CAP and by another accrediting organization called FACT, which is the Foundation for the Accreditation in Cellular Therapy. And basically what this means is that this is what's required for the FDA to allow us to process and manufacture various types of cellular therapies, such as bone marrow, peripheral blood stem cells, and CAR-T cells, and others. So you can see here we have we have an administrative area where our technologists sit, and then we have three clean rooms right here. Here are ultra-low liquid nitrogen freezers. And currently we have 7 technologists, a supervisor, a quality manager, and 2 directors. And in terms of facilities, we're located next to the Infectious Disease Diagnostics Lab, or the IDDL. Inside of our support area, we have a hematology analyzer, a flow cytometer, and 3 controlled rate freezers, which we use to cryopreserve products in a way that preserves viability of the cells. And we have several automated cell processing machines, about which I'm going to talk a bunch more later. We process between 200 to 300 products per year to treat patients with various malignant and non-malignant diseases. And we're involved in 27 active cell therapy clinical trials with various commercial and non-commercial sponsors. And so I briefly talked about how we have hematology analyzers and flow cytometers. Now, we perform testing, but we perform testing on products, not on patient samples. So this is a little bit different than the other clinical laboratories. So our tests will not appear in the EPIC results tab, for example, and we don't perform tests on patient samples. Now I want to talk about pediatric leukemia. So B-cell leukemia is the most common cancer in children. And survival of B-cell acute lymphoblastic leukemia, or B-ALL, is actually pretty good, approaches 90% in children. And so for about 80% of patients, they'll be treated with chemotherapy alone, and that is curative, so they won't have the leukemia come back. So overall, pediatric BLL does not have a terrible prognosis. However, if you are in the remaining 20% of patients who have relapsed disease, or who are primary refractory, meaning that the chemotherapy didn't actually clear the leukemia, the prognosis is much worse. And traditionally or historically, only about 1 in 3 of these children would have survived long-term survival, would survive their leukemia. So in the past, this is up until about 5 years ago, the traditional approach to relapsed or refractory leukemia was intensive chemotherapy followed by allogeneic hematopoietic stem cell transplant. And even though patients would receive this treatment, they wouldn't always survive it because the leukemia might come back, or the patient wouldn't survive the stem cell transplant, because that's a procedure that has a lot of morbidity and mortality. But all of this really changed for this specific patient population, for kids with relapsed refractory leukemia, through the advent of CAR-T cell therapy 10 years ago. This has really shifted the landscape for children with relapsed or refractory BLL. And to drive this point home, I want to introduce the case of Emily Whitehead. So Emily was 6 years old when she came to CHOP in 2012. She had relapsed BLL that wasn't responding to chemotherapy, and her disease was so bad that she wasn't eligible for bone marrow transplant. She was too sick to be considered for the kind of chemotherapy that is required before bone marrow transplant. And so she was really out of options at this time. And but exactly at this time, Steve Grubb's group was about to open an experimental protocol for a product called CTL019, which is a CD19-directed CAR-T cell therapy. And she was the first patient, the first pediatric patient who received CAR-T cell therapy. And she received these CAR-T cells and actually developed a really severe inflammatory reaction to CAR-T cells. We now, this is a side effect that we now know as cytokine release syndrome, and got very sick, had a very high fever, had to be transferred to the intensive care unit. And but when the fever subsided, when all this went away, she woke up and the leukemia was gone, and it hasn't come back. So in March of last year, in March of 2022, Emily was 10 years out from CAR-T cell treatment, and she's now considered the first patient cured by CAR-T cell therapy. So what is a CAR-T cell? A CAR-T cell is a T cell, which I'm showing here in blue, that's been genetically manipulated to produce a chimeric antigen receptor, or CAR, which is shown here in purple. And the CAR allows it to recognize and bind to a target antigen that's expressed on a tumor cell, which is here in the target is here in red and the tumor cell in orange. And when that happens, it can kill that tumor cell. So the extracellular component of the CAR is usually derived from a monoclonal antibody, from the antigen binding domain, antigen recognition domain of that antibody. And the intracellular component, the part that sticks inside of the T cell, consists of a signaling domain that activates the T cell. So this is called a chimeric antigen receptor because it's a chimeric protein. It's composed from two completely different proteins from an antibody and some signaling domains, and it's an antigen receptor because it can bind to an antigen. So that's why this protein is called a chimeric antigen receptor. Now, how do you get the CAR into the T cell? So we use lentiviral vectors to transduce T cells, but there are other approaches that use non-viral methods, such as transfection or electroporation of plasmids, to introduce the genetic material that will express the chimeric antigen receptor. CD19 directed CAR-T therapy works like this. So CD19 is expressed on normal B cells, from early development until their differentiation into plasma cells. And the expression of CD19 is restricted to B cells, meaning that there's no CD19 on blood stem cells from which B cells are ultimately derived, or in any other tissues. CD19 is also retained by most B cell lymphomas and leukemias, like ALL. So most when a B cell turns into a tumor cell, it usually doesn't lose the expression of CD19. And a really important consideration for immunotherapy against CD19 is that you can actually live without having any B cells. For example, Emily Whitehead does not have any B cells. So what do you need B cells for? B cells make antibodies, but you can get antibodies through an infusion of intravenous immunoglobulin. And so patients who've received CD19-directed CAR-T cell therapy, like Emily Whitehead, they'll come in once a month to get an IVIG infusion, and this is how they get their antibody. But the point is, you can live without B cells and survive. And so together, the fact that you can together this makes CD19 an ideal target for immunotherapy because there is little risk of autoimmune disease as a side effect of the immunotherapy. So CHOP and other groups have shown that CD19-directed CAR-T cell therapy is safe and leads to remission in the large majority, in up to 90% of children with relapsed or refractory B-ALL, with durable remission in some patients. We don't really know what the true rate of cure is because we're only now reaching the 10-year mark since we treated the first patients. There are two notable side effects to CAR-T cell therapy. The first I already talked about briefly in Emily's case. The first is called cytokine release syndrome, and that is an inflammatory condition that is due to the T cells encountering a lot of target cells and getting activated and becoming and starting to secrete a lot of cytokines, and this can lead to fevers as the first symptom, but can actually progress to multi-organ failure, and so patients can sometimes end up in the intensive care unit. There haven't been any cases of cytokine release syndrome in children that led to that were fatal. And typically, once the cytokine release syndrome is over, it doesn't leave any residual disease. So this can be severe, but is usually reversible. The same is true for the other key side effect of common side effect of CAR-T cells, neurotoxicity. So this can take any form. For example, confusion or delirium or behavioral abnormalities. Again, these can be severe, but are usually reversible. So the first treatment, the first time we treated a patient with CAR-T cells was in 2012. And based on this trial and multiple additional multi-center trials, the first CD19-directed CAR-T cell product that was approved by the FDA is tisagenlecleucel, or Tiza-cel as people usually call it, brand name Kymriah, and it's got a FDA approved in 2017. There are now 6 FDA-approved CAR-T cell therapies. We have Tiza-cel, Axi-cel, Brexi-cel, Liza-cel, Ide-cel, and Sila-cel. These four of these CAR-T cell therapies are directed against CD19, as you can see. So they are really similar to tisagenlecleucel. And among those, actually among all the CAR-T cell therapies, the only one that's approved for children is tisagenlecleucel. There are two approved CAR-T cell therapies for multiple myeloma that target a molecule called BCMA, or the B-cell maturation antigen, which is expressed on myeloma cells. Myeloma doesn't occur in children, so these two therapies aren't used at CHOP. At CHOP, we have many active clinical trials that use experimental CAR-T cell therapies for additional indications, including acute myeloid leukemia, Hodgkin disease, non-Hodgkin lymphomas, T-cell acute lymphoblastic leukemia, neuroblastoma, and other solid tumors. And more are in the pipeline. So, and I just wanted to introduce the idea of humanized CARs, because this is the flavor of CAR receptor that we're using in our current clinical trial. So what's a humanized chimeric antigen receptor? So some patients who get CD19 CAR-T therapy lose their CAR-T cells over time. So the CAR-T cells, for some reason, peter out. And one possible reason for why this might happen is because the antibody part, the external part of the CAR, is derived from a mouse monoclonal antibody. And those mouse sequences could lead to immune elimination of the CAR-T cells, for example, by the elaboration, by the emergence of human anti-mouse antibodies. So, but it's possible to make a humanized version of this originally murine antibody sequence by making just a few changes in the amino acids of the chimeric antigen receptor to make it then more similar to the human sequence. And the idea is that by making these changes, it'll be less likely that there'll be immune-mediated elimination of CAR-T cells, or cells that express this CAR receptor. And for that reason, the CAR-T cells might persist longer. But other than these few amino acid changes, the CAR molecule is really the same. It has the same target. We're still targeting CD19. It's really quite a small change. We have assessed such a humanized version of the CD19 CAR that was in a phase 1 clinical trial. This was Regina Myers, one of the assistant professors in our oncology program. And we saw clinical success in patients who had previously treated with CD19 CAR-T cells. So this is now, HuCART19 is now our preferred CD19 CAR, and this is what we use in the clinical trial that I'll be discussing in a short while. So let me switch gears now and talk about the process of manufacturing a clinical CAR-T product that is suitable for infusion into a patient and that's manufactured at a clinical scale. Here's a flowchart for what I'll call the traditional process of manufacturing CAR-T cells for clinical use. And by traditional, I mean really the process that all of the currently approved CAR-T cell products use to make the CAR-T cells. So the way it works is like this. You start with T cells that have been collected from the patient by apheresis. Then you purify or select or purify the T cells. The next step is to activate the T cell. I'm going to go into detail of how this works, but the idea is that you stimulate these cells to start growing, and this makes it this starts the cultivation process and makes them more amenable to gene manipulation, to transduction. Transduction is the next step, and after transduction, there is a cultivation period that goes from 8 to 10 days, during which the cells grow by between 10 and 50-fold from the starting material. Finally, the cells are harvested. They are formulated in a final formulation buffer, usually consisting of saline and albumin. So this is what they'll be suspended in during infusion, and then the cells are ready for cryopreservation. We usually like to infuse, while it's possible to infuse CAR-T cells fresh, we prefer cryopreserving them, because that really uncouples the logistics of manufacturing from the logistics of infusion, and that makes things a lot easier. Now, what I wanted to show here is that there's a lot of different machines and a lot of different bags involved in doing this manufacturing, and you need highly trained technologists to perform these manual steps during the cultivation period. And to address this, there's now a trend to manufacture CAR-T cells in so-called GMP-in-a-Box systems. And what's a GMP-in-a-Box system? These are benchtop machines that could perform almost all of these steps that would normally or traditionally be performed in different machines and separate devices. You perform all of these steps in one machine and in an automated fashion. The Clinimax Prodigy is one such GMP-in-the-Box device, and here's what it looks like. You can see that there is a computer, and this is an electromagnet which can be used to purify cells by magnetic separation. And you can see various bags that are connected to a tubing set. A tubing set is a little bit difficult to see here, but maybe you can see these tubes running all over the surface of the machine. The tubing set is closed, and this minimizes the risk of contamination. And it's also disposable, which means that after manufacturing is done, there's no need to decontaminate the device for changeover. You simply pull off the tubing set and all of the connected bags and throw everything in the biohazard trash. From a technologist's point of view, the manufacturing process on this machine, or on this type of machine, is pretty straightforward. The first step is you install the tubing set. Then you sterile weld the bag with the T cells to the tubing set, and also bags with various reagents such as lentivirus and media and buffer and other such things. Then you set the program on the computer, and after this, manufacturing proceeds mostly automatically. Now, throughout the process, you can take quality control samples to do things such as monitoring the growth and the health of the culture. And at the end, you'll have a CAR-T cell product that's ready for infusion or cryopreservation. So to recap, GMP-in-a-Box CAR-T manufacturing proceeds in a functionally closed, sterile point-of-care system in a decentralized fashion. The process uses an automated cell processor that can fractionate, wash, transduce, and culture products at a clinical scale. It uses there's a single benchtop device that replaces incubators, cell rockers, and refrigerators and other devices. And importantly, the process is compatible with the kinds of reagents that you'd use in traditional manufacturing. So you can use the same media, you can use the same lentiviral vectors. So you can really switch from the traditional to the second generation manufacturing approach. So the next couple of slides, I want to outline a clinical trial that we just started, recently, that uses Prodigy manufactured humanized CD19 CAR-T cells, or HuCART19 cells, to treat pediatric BLL. So this trial design is very similar to the kind of clinical trials that were done for the initial approval of CAR-T cells. So these are just a similar trial than the one that Emily Whitehead was on. But I want to talk you through this in some detail, just to give you sort of a sense for how these trials that we do at CHOP, how they're set up and how they work. So the point of this trial was to test the safety and efficacy of taking the HuCART19 cells, HuCART19 vector, which is our second generation CAR-19 vector, and moving it to a second generation manufacturing process using the Prodigy. And there's two phases to this trial. The first phase, phase one, is a safety, and this is being done using what's called a 3+3 trial design. And it's a simple way to think about this is that you want to try two different dose levels, one that's slightly lower than what you think is the ideal dose, and then you enroll 3 consecutive patients at the lower dose level, and if you don't see any dose-limiting toxicities, then you can move on to the second dose level, and you treat 3 additional patients. And then if you didn't see any dose-limiting toxicities in these first 6 patients, you're done. And if you do, then it becomes a little bit more complicated. But this means like after 6 patients, the phase 1 arm can be done, and you can move to phase 2. And we have many more patients in the phase 2. The phase 2 trial, or phase 2 part of the trial, is meant to measure the efficacy of these HuCART19 cells. And we define efficacy as the overall remission rate at day 28. So importantly, this is not the same as cure, but we won't know whether patients are cured for many years. So we need to have an early signal to know whether the CAR-T cells are working. And this is done by looking at disease burden at day 28. And there are secondary aims, secondary endpoints, such as feasibility to manufacture, our ability to manufacture these cells. We're going to look at safety. Importantly, we're going to look at in vivo survival of CAR-T cells, as well as measuring long-term event-free, relapse-free, and overall survival of patients. So here's the study population. We intend to enroll 89 subjects, children and young adults, for a target of 80 infused subjects. And we're including patients with CD19 positive BLL or lymphoma that is relapsed or refractory, and whether or not, and we'll also enroll patients who have this and have had previous treatment with B-cell directed engineered cell therapy, which really just means they've had CAR-T cells before, and the CAR-T cells didn't work. There's 2 cohorts to this study. The first cohort is patients, cohort A is patients that have relapsed or refractory BLL or B lymphocytic lymphoma, CD19 positive. And the cohort B are patients who also have this, but who've been previously treated with CAR-T cells. So cohort A is CAR-T naive patients. Cohort B is patients who've previously had CAR-T cells. And we're making, and then in the two phases, in the first phase, we'll only enroll patients from cohort B. And the reasoning here is that these patients are sicker, they have fewer options, and so it's more appropriate to determine the safety profile on patients who already have fewer options at this point. However, once we establish safety in the phase 1 part, we'll move on to phase 2, where we'll enroll both cohorts. So here's the study schema. Patients are consented and enrolled, and then we collect T cells by apheresis. CAR-T cells are then manufactured using the Prodigy system, and once all of the release tests pass, and we'll talk about release tests a little bit later, patients undergo lymphodepleting chemotherapy. So why do you give chemotherapy before CAR-T infusion if the CAR-T cells should be able to deal with leukemic cells? So there's two reasons for this. The first is that you'll want to open up niches in the bone marrow and in the lymphoid organs to allow the CAR-T cells to engraft. The second reason is that when there's a lot of tumor burden at the time of CAR-T cell infusion, you'll have a much greater risk for severe cytokine release syndrome. And this makes sense because if you think about it, if you have all these CAR-T cells that are active and now they see all of these CD19 targets, they'll start dividing and getting really activated and start secreting cytokines. Whereas if, on the other hand, you don't have a lot of target cells, you don't have a lot of tumor cells, this is the CAR-T cells are not going to do that. So that's why we do lymphodepleting chemo, and then followed by the infusion of HuCART19 cells. Here are the two dose levels. The first is 2 million CAR-T cells per kilogram, and the second dose level is 5 million CAR-T cells per kilogram. And then we evaluate patients for disease response with a bone marrow biopsy and a lumbar puncture at day 28, and this is where we're gonna determine whether there's residual disease. Follow-up then continues for a year, and then if at the end of 12 months, patients get enrolled into a 15-year long-term follow-up study. So we really want to keep track of these patients and their long-term outcomes after CAR-T therapy. And at this point, we have successfully manufactured 6 patient products and infused 3. So far, we've achieved 100% complete response rate at 28 days. Well, this is from 2 products, but this is looking good so far. So an important aspect of cell therapies is what's called release testing. So what's release testing? Release testing are FDA-mandated clinical laboratory tests that the product needs to pass before it gets infused. And these tests are meant to verify the identity, the purity, the potency, and the safety of the product. So here are the release tests that for the Prodigy HuCART19. And by the way, an important part of writing an application for an investigational new drug, or an IND, which is what we had to do to get the FDA to approve this clinical trial for us, is to negotiate a proper release testing and specifications. So this is the release testing that we have for the Prodigy HuCART19 cells, um, and dose calculation is really important, as you might imagine. So to calculate dosing, we use a hematology analyzer to count the total nucleated cells, or TNC, and then we multiply this by the percent of TNCs that are positive for the chimeric antigen receptor using flow cytometry. Cell viability is an important attribute, especially after cryopreservation, and we measure this by flow cytometry for 7-AAD. We also use flow to measure the purity of CD3 T cells and the presence of residual leukemic cells. So remember that, so why are there leukemic cells in the product? So remember that the cellular material gets actually collected from the patient themselves. So the patient has leukemia. So this means that there could be some leukemic cells inside of the product, and if we infuse that product back to the patient, we wouldn't give them any leukemia that they didn't already have. However, the FDA wants us to make sure that fewer than 1% of the cells that we infuse are actually leukemic cells, and this is usually not a problem. This is an easy bar for us to take, because first of all, we do purify our T cells before we manufacture CAR T cells, and then even if any of them slip through the purification process during cultivation, the CAR T cells should actually be killing these cells. So less than 1% is fine, and we usually don't detect any. Most of the other tests verify that the product isn't contaminated. So we perform endotoxin testing using a handheld point-of-care device, and we measure mycoplasma by quantitative PCR. And this is done in collaboration with the Infectious Disease Diagnostics Lab, and I think this is a really nice example of interdepartmental collaboration. And I want to give a shout out to Mike Elkin and Kara Thompson for facilitating this for us. Sterility testing is done using an FDA-approved method that takes 14 days to finish, and this is done in a commercial outside lab. And for lentiviral therapies, the FDA also wants you to look at the number of vector copies that are integrated into the genome, and they want you to look for replication-competent lentivirus. And these are tests that we farm out to Penn. So in summary, pediatric leukemia can be successfully treated with CD19-directed CAR-T cells. We learned that a CAR-T cell is a T cell that's been genetically modified to express a CAR molecule, which helps the cell detect and kill target cells. And we can manufacture CAR-T cells using GMP-in-a-Box automated cell processors. And then, importantly, release testing ensures that the CAR-T product, or really any cellular product, is pure, potent, and safe, and must be finished prior to infusion, and is and highlights an important touch point between cell therapy manufacturing and the clinical labs. So I want to acknowledge the Cell and Gene Therapy Lab team that has developed the CAR-T manufacturing process, and the clinical team led by Steve Grupp. And funding for this clinical trial was provided by the CHOP Cell and Gene Therapy Collaborative.